So yes, I'm going to talk about species interactions, and I gave a Alien CSI webinar. It was only a couple of months ago or something like that. At least it feels not that long ago. And many of these slides are from there. But I want to sort of talk a little bit differently about species interactions, specifically for this this meeting, so that it just makes us think a little bit different about how we can approach different citizen science projects for collecting species interaction data. And this was the title I got, but that's very appropriate to what I want to talk about. You do see my slides, I assume. Yeah, great.
Yeah. We can see everything. Don't worry.
So iNaturalist, I'm sorry to Alexis and Pierre, but I use iNaturalist a lot. But I thought I'd use them as an example of very traditional, in actual fact, recording, because people go on there and they collect their stamp collecting species in many respects. And I was just on a field work last week in Spain or Catalonia, sorry, Bernard. And I was with a young colleague of mine, and it was quite interesting to see him basically stamp collecting species as he went around. And if you look at iNaturalist, you have their classical visualizations that you get on all of these. So if I'm going to go straight to iNaturalist, and I can create visualizations like this, I can see all of the lizards and reptiles I've seen, for instance. And we shouldn't forget that these kind of assemblies of pictures are some of the visualizations we can get from these kind of apps. And you know, it surprises myself, and I can't even remember myself seeing half of these different species. And of course, the classic one is the maps, and we still use maps a lot. I guess they started being produced with computers in the sort of 1960s. And have come a long way since. And so we have maps like this of any particular species, which of course we know isn't really the distribution of that particular species. It's a combination of the distribution and of the activity of people. But maps have also evolved because I can zoom into them, and I can see individual records, and I can click on those records, and I can view that particular record. I can then decide if that I agree with that identification and things like this. So maps are not quite what they used to be, but have advanced. And we've also got phenology, which iNaturalist does, and you can see it loading here. I picked Arum maculatum for this example because it's classically double peaked, because it has a peak when it flowers in the springtime, and it has a peak when the bright red berries are obvious in the autumn. This is so this is an actual phenology of detectability of Arum maculatum. But if I click on the phenology here, it should load the red lines down here when it's flowering, and the blue line there is when it's fruiting, and you can see the separate thing. But again, that's a combination of the detectability and the actual plant flowering or fruiting. So this stamp collecting doesn't tell us anything about ecology, really. It tells a lot about biodiversity in particular places, and we know from our knowledge of biology that things do interact. And yet, traditionally, people haven't gone out and collected interaction data. And we have to think, how can we encourage this? Because if you really want to use these data for ecology, there's an enormous number, and I'll get onto a bit about the questions, enormous number of questions we could ask about the different interactions between species. And of course, humans are at the sort of, I wouldn't say the center of ecology, but humans are part of this network. And so that should give us a link between people and all of the different organisms involved. So yeah, I started listing a few of the questions that you can think to to ask about the interactions of different species. So obviously, what species interact with other species, so the range of different hosts, parasites, pollinators, etc. But where do they interact? So things don't necessarily always interact if they're present. They might interact at different particular times of day, at particular times of year, things like that. How often do they interact? Are they very fussy about what they interact with? You can imagine in different places things interact with different things. Is there competition for those interactions? So competition for prey species, for instance, and what are the range of different interactions that an organism has? And potentially, though, all of those different questions could be answered by citizen science, but we need to find ways to motivate citizens to perhaps collect those kind of data. So let me just take you to Globi. So this is a global biotic interactions database, which is just a big global aggregator, and it's run by Jorrit Polen. And it just links lots of different data. And I thought I'd take you there because it shows some visualizations of interactions. So I'll just use the example of he has up there of honeybees, and you can click on it, or you can fill out this, and you can just get a long list of all of the interactions that honeybees have. That's not quite a visualization; it's more of a list. But you can also use the browse function, and it has the example of this. I think there's some sort of turtle in there, but you can add anything you want, and it has a nice. There's a couple of visualizations here. This one is probably the most useful. And you can see there's the turtle, the sea turtle there, and all of the different species that it interacts with. And I think the color is significant to what kind of organism it is. I think green are plants or algae in this particular case. I look at it, and I suspect some of these black ones are things like barnacles and other stuff. I'm not much of a marine biologist, so I don't know much about turtles. So that's one visual. Sorry?
Sorry, I thought I was on mute. Tiger sharks, the turtles interact with those.
Ah, okay. Yes, indeed. Not in a good way, I imagine. So that gives you some idea of just the range of species that a species interacts with, but it can't start at the moment to really give you that information about when and where and things like that. So as Tim in his early introduction said, I worked on a script, it's almost 2 years ago now, my God, still needs publishing, which combined Globi data with GBIF data to create visualizations of interactions. So if I click on here, we'll actually go to the dynamic interaction. And I talked about these before. Many of them, I'm sure you've heard this before, but the circle size is related to the occupancy, in this case in Belgium, of that particular species. And the green means plant, the blue means it's an animal, and it has various different interactions. So Vanessa Atalanta, which is a butterfly, eats, and in this case it's grapes. So it doesn't eat the whole grape. I think it just lands on the grape and sucks juice from damaged grapes, I imagine. And I made this particular network of Vespa velutina, and I used the interaction network of Vespa velutina for 2 reasons. One is Vespa velutina is not a massive eater of grapes. It apparently visits the flowers of fennel, and apparently it also eats Fatsia japonica. Well, in actual fact, it probably just visits the flower. But we know the Vespa velutina eats many more things than this. And what is really showing you this network is our ignorance of what Vespa velutina does eat when it comes to Europe. There's probably a lot more information had I done this in Japan or Southeast Asia in general. The other thing I use this why I use Vespa velutina is if you look at an example where of a common species, if you took a roe deer, for instance, then the whole network would be so incredibly complicated, you wouldn't be able to figure out anything from it. There's just too many interacting species to make sense of it. And so these visualizations are quite nice, but there's still a lot of work that needs to be done to make to make these attractive to somebody who's trying to explore and understand the network. So let's go back to here. So in terms of interactions, there's still a lot of work to be done to make these data open, and it's not just a matter of making them sort of openly licensed. It's a lot of these data are hidden away in supplementary data to sort of pollination papers and things like this, and they're just, it's not that the people want to hide them or anything like this, it's just they don't know where to put them to make them more open and standardized so that we can share those data easily. And so there is work going on in the standardization of species interaction data, but it's not easy, and different interactions have different demands for standardization. These networks that I made take a long time to make, and so there's an element of the efficiency that platforms would need to be to make these on the fly, for instance, and making them easy to browse and understand, that we mentioned. And also there's a lot of statistics you can do on networks, but to make that interesting to a citizen science, that kind of has to be explained. So it's easy to say that this network has or this node has a certain degree, but what does that mean to somebody? Or how long does it take to transverse a network? What does that mean to somebody, and how can you explain it to a citizen who's maybe trying to explore the networks? And yeah, and in general, how we use these data, how can we incorporate these data into our models and make much more use out of them? And that's still a lot of work that needs to be done in that area too. So going back to iNaturalist, if I go back again, so iNaturalist does sort of collect these data, but it doesn't use them. So if I, for instance, I'll filter it on, I'll go to my observations. I'll filter it on insects. It's an insect. And I should be able to find something sitting on a flower. There's a beetle sitting on a flower, but I don't recognize the flower. So there's what's most identifiable? I'm sure I saw one of these in it earlier. Yes, there we go. So there's a marmalade hoverfly sitting on some sort of wild rose. And down the bottom here, it says observation fields, and I can say, I can choose flower visited, and I can put in Rosa. And I can add it. And that will sit there and nothing will ever happen to that, except it does get harvested by Globy. And so those data go in Globy. But if you imagine if every picture of the marmalade hoverfly was annotated like that, you could get all the ranges of flowers that the marmalade hoverfly visits, the times of day that happens, the places where that happens, and be able to compare that with the occurrence of those two different species and things like this. So iNaturalist has the tools to potentially build networks and display networks, but at the moment it's not doing it. And so I would hope to encourage all of these platforms start doing that, and then hopefully that would encourage be a sort of cyclic thing that encourages more people to do it, because then they can see their interaction coming into the interaction network, if we can make it suitably viewable, that it doesn't get too complicated. Our very last physical meeting from Alien CSI was hosted by Honza in Pruonica. Seems like a long time ago, but we discussed species interactions a lot in that meeting, or at least one group of us did. And we thought a lot about how we would try and encourage citizen science to collect those kind of data, and eliciting emotional connection to the organisms was very important to that. Keeping it simple and fun, which is a common thing within citizen science. And also providing tools for citizens to make visualizations of networks, we felt was quite important as well. So if you think about the different kinds of interactions that there are, how can you make these interesting? I would say it's kind of difficult to run a citizen science project on what barnacles are on the back of my turtle. But you could potentially think of some of the symbiosis. Predation, there's a project on iNaturalist to do with sea otters in California and what they eat. I'm sure I've seen a project about what the cat dragged in terms of domestic cats preying on organisms, and there's definitely scope for that kind of project. Herbivory is something you could work on with gardeners, for example, who would be very interested in what's eating their vegetables. Disease, in this case, I've pictured a plant disease, again, gardeners potentially, but maybe others. Things like ash dieback and things like this have been important in Europe recently. And maybe there's other pests as well. Pollination, I know Jody and Helen and Kelly have been working on the Pomsky project in Cyprus, and they will have to tell you how successful that is. But I think there's a lot of scope for pollination and potentially to elicit the positive responses towards insects that way, and flowers as well. And let's not forget vectors, because that was a big conversation we had in Puonetso as well. We particularly were talking about mosquitoes, and how we could collect data on mosquitoes from citizen science and the different sorts of things we could collect about mosquitoes. So when they bite, where they bite, what environmental conditions were correlated with biting, where are mosquito larvae found, for instance, and all sorts of other data that potentially is very easy for citizens to collect in their own homes if they're given sufficient motivation to do it. And we have all these other hypotheses we could think of working on to do with invasive species, and I'll just go for those because I'm sure I've taken up too much time already. But we can think about all of those different things if we start collecting the data. So extending citizen science platforms, as I said, I think we're halfway there. We just need to go the last mile to do it. There's a lot we can do by combining individual observations of single organisms with interaction data from elsewhere. But there's a lot of work we need to do to try and make this put in an understandable format. And I think that's really it. But yeah, that last point about using the unique features of citizen science is very important. Things like the mosquito data allows you to get within people's homes if they're willing. Not necessarily located, but it allows you to do biology and species interactions and species identification in places that scientists just can't get. And so citizen science could have a very significant role there. So I think that's it.
Thank you very much, Quentin. Thank you. It was a really interesting, a great presentation too. Yes, I have to say also very interested in this interaction issue because it's challenging, but it's really fascinating. Yeah, and Alexis was commenting that some interaction are easier to be observed by citizen science than others. Like interaction between plants, for instance, are difficult to be observed. Yeah.
Well, absolutely.
Yeah. Think you agree?
I mean, things that happen at night, things that happen in rather yucky habitats, like underwater and things like this. Yeah, you have to choose your target, very much so.
Yeah. I'm very curious to know what's happening under the water, but it's not always easy to see too. So Ellen is commenting and excited to think about the ways in which technologies could assist in capturing less visible interaction, and maybe like changes in semiochemistry when interaction occurs. So other issue, other aspect of this. So
I was thinking about some of the technologies like the eNose technology, or I mean, maybe underwater there's some kind of acoustic technologies that could pick up changes in sound as a consequence, for example, of predation events. I don't know, hearing them scream, I guess, is what I was thinking in that sense. But
no, you're right.
And yeah, so I think anything is possible basically, but it's really inspiring to hear Quentin's ideas and, you know, thinking about simple things like that annotation within iNaturalist that could really make a big difference going forward. That's really seems to me something that could be quite achievable if more people knew about it, and if there was some as well some kind of proofs of concepts of the ways in which that could be used. And it was really fun working on the paper with Quentin and others. So thank you for that opportunity. And
we've still got some work to do.
Yeah, almost there though. It's been,
Almost.
yeah, I've just got the revisions back. So that's good news. And yeah, thanks to Honza for hosting us and getting that work going.
Yeah. Before moving to Stefan, Tim asked if there are many interaction data that can be inferred from pictures already. Are there any tools we can think of to harvest this? Do you have any idea, Quentin?
I've talked about this a lot, but I haven't actually done anything, and I think it's a work in progress. I imagine some people are doing it. I know, Tom, did you ever manage to do anything along those lines? Oh, sorry, I caught Tom out now.
You have caught me out. Can you say that again?
Yeah, we talked like 2 years ago about trying to identify the plants that an insect was sitting on.
Yeah, no, I didn't take that any further.
No, we talked a lot about it, but likewise, I haven't done anything. But I don't know if anyone else here has.
So there were exciting ideas, weren't there, around the kind of combining the image analysis side for both the insects and the plant, but your work in South Africa as well, Quentin, was really exciting in terms of those systems that you can already use to join things up.
Yeah, absolutely. Still a work in progress, and I really like those were very much about combining known sorts of interactions with sort of current data on occupancy and things. But the idea that if you have enough of this data, you could start looking at the phenology and more detailed things about the actual interaction sounds quite exciting, really, and could reveal things we just didn't know, because these interactions are very fleeting and you need a lot of data from lots of different places to be able to discover some of the things we don't know about interactions.
Thank you, Quentin. Stefan.
Yeah, this is really fascinating. It's interesting to think how much data is at our fingertips and we're not actually using it, and it's already there in a sense. I'm wondering, some of the examples you showed, there was sort of, you know, after the fact. So there is data and you could pick up additional information from that, like with this example from iNaturalist that you used. I'm wondering if you thought about how that could be built into a citizen science project from the start to collect this kind of information, and also what kind of trade-off that would mean, because building that in would make maybe the process and the workflow more complex, and then it might actually impact the participation or the level of participation. So I'm wondering if you've thought about this.
Yeah, I mean, that's the significant challenge. Once you start making it too complicated, you get a great dropout rate, and so it's finding that fine balance between the two. Yeah, we thought about it. I'm not sure we came out with it. I think eliciting this emotional response is very important, but getting the stamp collecting kind of bug on this would be really helpful, because that kind of I don't know, the way people just go looking for as many different species as they possibly can in an area, trying to get that going on interactions, I find difficult to imagine. But if we can figure out a way that you get that kind of response to a website or what have you, then that would be fantastic. I meant to mention, actually, I had a MSc student who wanted to do a little project, and it was right at the beginning of COVID when she came along. And I suggested to her that she goes through iNaturalist pictures of bats and randomly selects, there's something like 40,000 pictures of bats of different species. And she looked at a random selection of, I can't remember how many we did, and saw whether the people were wearing gloves or not. And then we plotted whether they were wearing gloves or not with time, to see whether having a pandemic going on made a big difference to glove wearing. What actually happened was glove wearing has gradually improved with time for the last 20 years, in actual fact. Because in actual fact, although iNaturalist hasn't been going for 20 years, people upload old pictures, and so you can actually get quite a big timeline. So we did this manually, but it made me think about using machine learning to detect gloves or not on bats. And what I found very interesting was that if you try to make a training data set to try and recognize people holding bats with gloves or people holding bats with bare hands, would you be able to do that? And I think the answer is it would be extremely difficult, because the detectability of gloves is not the same as the detectability of nice pink hands. Because gloves can be black, they can be blue, and things like this, and they can be hidden in the background. And so not everything, a human, obviously there's going to be a slight detection difference, but for a human detecting whether the bat is being held by gloves, but I don't think it's as big or as dangerous as trying to do it via machine learning. And I'd be quite interested to know, because obviously you'd then have a bias in your data set created by the machine learning. And I don't know how much we've addressed that kind of bias and the dangers of that kind of bias in our data sets. So that's a bit of an aside.
Thank you, Quentin. The team has a couple of questions. So, to what extent do we have good standards for ecological interaction that citizen science platform could use? He commented that some people, that in the Belgian recording system, sometimes people get the comment, please do not post pics of multiple taxa, because this creates problem for the automated image analysis, identification. So it could be a shame, because we can lose a lot of data.
Yeah, well, I can't agree more. I mean, and obviously, so what? Sorry, I'm forgetting his name. Milan, who did a short-term scientific mission here, right in the first year of Alien CSI, looked at pictures of bees, butterflies, and hoverflies, and found, at least for bees, it was something like 60% of all bee pictures were on a flower. And I think it was 40% for butterflies and moths. So at least in those cases, if you exclude the ones that don't have another species in them, then you're throwing out an enormous amount, and you're throwing out all those interactions as well.
And I was wondering, looking at your graphs, for instance, and you gave the example of the butterfly that eats a certain plant. Are there any good standards actually, or is it just a mass of ecological interactions that you could potentially use in a citizen science platform, or is there like a quite well-defined suite of ecological interactions with clear definitions that?
There's the, I'm trying to think of the name of the ontology. I've forgotten it. There is an ontology that's used in Globi, which is called the, no, it's gone. So that's just to find the interactions as such. But I wouldn't say it's complete. We recently got co-roosting put in there because species co-roost together, and apparently co-roosting was not seen as an interaction before recently, but there's probably other interactions that are missing. In terms of the data, there are many different ways and there's many different considerations. Pollination is very, very different to predation in all this metadata. And you can think of them as two different occurrences and linked by an interaction. But then you are missing some of the details that way. And there is a group under the Biodiversity Information Standards Organization that's working on better standards. And I believe they have a workshop at the next meeting, which is in October. So please do come along. I'll put a link in the chat.
Thank you, Quentin. Okay, I think we have to move to the next talk. I just want to mention that there are very interesting comments in the chat, like the one raised by Pierre about collaboration between the people network who study plant pollinators interaction. That in this project, citizens identify insect species and they use plant and service for species identification. So it would be interesting, Pierre, if you can send us a link or something explaining better this collaboration, and also now reminded that in the future there are new cars like Tesla with tens of high quality cameras that are constantly looking surroundings, so they can be a source of data. And other comments. Okay, and Quentin put the link for the conference. Thank you very much. And thank you very much again, Quentin, for the very interesting talk. So a lot of comments and questions. So people are really interested about this topic. And now it's the turn of Jim, who we talk about wildlife cameras.